I'm Liam Fanning, and I'm from Cork in Ireland, the southwest coast of Ireland, and this is a relatively new area of research for my laboratory. Just to give you some kind of context, I used to work on hepatitis C, HIV, and do a lot of host pathogen engagement, looking at kind of signatures and genomes for with regard to cytotoxic T cell escape, and then looking at some signatures of humoral immune escape. And I suppose thanks to, we'll say, the direct acting antivirals, which were kind of very effective at offering curative therapy for hepatitis C, and the funding dried up, I had to kind of pivot, and working in a laboratory where we had some very eminent gastroenterologists, the obvious kind of connection there with looking at the gut, bacteria, the microbiome, and bacteriophage was an obvious lead for me to apply what I knew about host pathogen engagement and to bacteriophage and viruses. So I'm part of the APC Microbiome Institute at UCC, and I'm operating under Theme 4, which is Host Microbiome Dialogue. Okay, so we don't seem to want to move. Right, why aren't we moving? Apologies, it seems to life of its own all of a sudden. So we have the IT guy up here just so that I'm making a mess of this. Doesn't seem to want to advance. Okay. So the important slide, I know it's left to the end, but I'm going to include it at the beginning. Oh. Oops, back share. And yeah. We seem to have, okay. Right, now let's hope that's the end of the snafus. This is the my collaborators. Particularly, I want to thank Angus Lavelle, who's the bioinformatician who does a lot of work on the biome data that I'm looking at. Aoife Sullivan, who's going to be presenting in the next talk on the human immune biome, and my other collaborators there, particularly the Hill Laboratory, who developed the bioinformatic pipeline that Angus uses, and Emma McDermott and NUIG for doing some of the EM stuff. Why aren't we advancing? Okay. So actually, in the interest of time, I'm going to skip that one. So what is an immune biome? Well, a biome is a naturally occurring community occupying a habitat, as we all know. An immune biome is any component of that nascent biome, which has been recognized by the immune system. And I suppose the microbiome and phagome have been very richly explored, and the immune biome largely unexplored. We are seeing some evidence in regard to kind of the IgA coating of bacteria and that association with colitis and that rich environment that happens when we have an inflammatory process of disruption to the IgA coating. And I'm focusing primarily today on the viral biome, but we can see that, you know, luminal bacteria coated IgA and IgG during intestinal inflammation, all that kind of disruption to the microbiome and colitis. We're seeing quite a rich portfolio of papers beginning to come out on it, but not so much looking at the actual specifics of the antibodies that are binding to them. So I've divided the immune biome kind of into adult and the perinatal and neonatal, and there are, if you like, three parts to this: B for biome, I for immune, and E for the enteric gut system. And as part of this ecosystem, obviously we have bacteriophage and phage, but we have a kind of a microenvironment where we're sampling immune cells, M cells, dendritic cells, seeing what the, if you like, antigen epitope portfolio is, and then providing, maybe responding with IgA and IgG antibodies. I suppose you could say, why do we have an immune biome, or what is the whole purpose of it? Well, from an evolutionary context, we've been, you know, living with bacteria and bacteriophage for certainly our ancestors anyway, for millions of years, and that it potentially induces, from an immunological perspective, systemic tolerance, and it probably on some level sets the predator-prey equilibrium in the gut ecosystem that we have, and the other organs, including the lung and so on. And the purpose is probably to maintain lifelong tolerance to these non-self biomes. They do contain the diversity and the richness of epitopes which have the potential to react to, and we don't want to react to them. That's probably what happens in some contexts of, we'll say, some of the inflammatory diseases. And it's also a means to enhance a mucosal immunity and balance again that predator-prey dynamic that I think is key to kind of maintaining maintaining a healthy ecosystem within our guts. How? Well, I'm only looking at the humoral immune system, but undoubtedly there'll be a kind of a T cell arm to this. But by coating biomes with IgG and IgA, they are signals that are sent, that are kind of interpreted within our mucosal immunity and our systemic immunity to determine how we react to these particular entities. And then when? Well, probably initially seeded in the perinatal and neonatal period, depending on the animal. As humans, you know, we get transfer of IgG across the placenta and some IgA. But if you, some of the animal models are really nice in this and fortuitous, they're born agammaglobulinemic, which means they have no antibodies, and they are really dependent on getting colostrum in order to get their first feed of immune molecules from the mother. And really in the adult then, I'm putting forward that there's kind of continuous sampling of the biome, and that we get this prospective change to the kind of diversity or antigen binding capacity of the IgA portfolio of antibodies, the IgG being kind of slightly more historical. So in the perinatal and the neonatal, we have colostrum as increasing this kind of tripartite axis to expand it to 4. And as I said, in humans and those kind of mammals, we have in utero transfer, but in the likes of calves, we have only transfer through colostrum. And this adds a kind of a new kind of thought process to kind of where all this immune biome seeding and education of the immune system comes in, and what's the importance of colostrum. So really, it's about preparing a functioning gut ecosystem that regulates predator-prey dynamics. And for the purposes of this talk, my animal model is the cow. In the hours post-delivery, we collected colostrum and feces in the 6 hours after delivery of the calf, and we're currently focusing on the humoral immune biome. And again, as I've said, why calves? Because they're born particularly with their agammaglobulinemic status. This is only one aspect of what my lab is looking at. We're looking at the immune biome in humans, and also we're beginning to look at it in non-human primates, 4 species in a collaboration with Dublin Zoo. So the workflow in order to kind of get to the next generation sequencing data is that, as I said, we harvest some of the colostrum. Cattle produce quite an amount of colostrum, so you know, getting 100 mils of this very valuable material is not an issue. We then, after removing the fat in the casing, we cesium chloride grading purified the phage. We do an ELISA on it, IgG or IgA, depending on what experiment we're doing. And then we do an enrichment process. We cesium chloride gradient again in order to kind of make sure that we've got as much free antibody as possible in the preparation. We do an IgA ELISA again to make sure that we're actually after enriching for the particular immune-associated phagome that we're looking at. Then we extract the DNA in this particular case, and we send it for next generation sequencing. And in this particular data set that I'm going to show you, when you got the data yesterday, so it's fresh off the press, so perhaps appreciate maybe not sharing some of the data. We had an N of 10 animals, 10 cows, and I'd like to acknowledge Bernie Crowley for doing this work, particularly on getting the DNA ready, up as far as the DNA ready. This is for those that are interested in the bioinformatics. In the interest of time, I won't go through it, but this is our, if you like, bioinformatic pipeline of positive, negative selection, looking at previous reference databases and trying to pull out those particular contigs that have greater than 3 PVOGs, and some predicted to be circular in the CRAS databases as well. So at the end of that, we end up with a bioinformatic contig, so a database of contigs, which Angus Devell has curated for us, and then we end up getting graphs like this, and this is where we start getting into the meat of looking at the detail of the phagome that is in the both the starting material, which is colostrum, and the kind of IgA enriched portion of that. And really what this graph is actually telling us is that there's really no difference between the diversity in the starting colostrum and in the IgA, and that while there's a very large degree of diversity and there's lots of phage in the IgA population, that's about as much as I can tell you at this first level alpha diversity analysis. With the beta diversity, again, what we can see is that usually, if you look at fecal material, each of the pairs would coalesce together. But here we're getting a little bit of spread in some of the individuals. But for the most part, there are no large-scale differences in the colostrum in the samples between the starting material and the IgG enriched samples at the community level. And what this is telling us is either one of two things: either that our enrichment process is actually saturated and we're not actually, we suspect that we're not pulling out all the IgA associated bacteriophage, and that what we're seeing is a proportional representation of those bacteriophage that are IgA associated. And looking at the beta diversity for each of the 10 colostrum samples that we looked at, most of them are clustering down in the bottom left-hand side there, the IgA proportion of the biomes, and that while there were large inter-individual differences, it didn't reach quite statistical significance, and this probably reflects the fact that we've only got 10 animals in the study, and we're hoping to power it up a bit more with some other animals. There's some population convergence at the IgA level there in particular, kind of 8 out of the 10 animals seem to have this kind of convergence around the PCOA plot, or the Bayes-Curtis diversity plot. And data that I'm not going to show you is that the colostrum diversity is different to the very large diversity that we see in fecal samples, and that there's less inter-animal variation in the colostrum samples than there is in the fecal samples. So from a taxonomical perspective, on the left-hand side, we have the data from the 10 samples from colostrum, and we have the IgA enriched proportion on the right-hand side. And for the most part, you can see that they're actually quite similar. But one thing to note is, particularly with the biome databases, is that they're very poorly annotated with respect to the human databases. And that's why you can see that in some of the samples, nearly over 60% of the contigs can't be assigned to a family group. And I suppose, we can see that we have poly-DNA viridia there as well, and plenty of other the representative ones. But what I'd like to draw your attention to is some of the animals on the right-hand side, particularly the last two, is that the IgA pull-out actually seems to kind of differentially select out a proportion of the bacteriophage compared to the starting material. And that's the kind of thing that some of the signals that we're beginning to look at and trying to kind of put some kind of meaning on those signals. For this particular plot, it's actually the log fold is going on the bottom of the screen from 0 up to 8. And what this is really showing is that there is some differential between the colostrum and the IgA enriched phage. In other words, that we're seeing there's more of these families in the raw colostrum than there are in the IgA enriched population. And what this might be is that it signals that the family differences are systemic across the cohort, and that we have a group of phage perhaps that are not IgA associated, and this might reflect the fact that they might be immune invisible, or less antigenic, and that therefore we mightn't be pulling them out in our IgA enrichment. The value of knowing this information is that, as you know, I suppose with COVID-19 we were using chimpanzee adenoviruses as vectors for particular kind of and that. So if we can find a group of we'll say bacteriophage that are immune and silent, in other words can't be seen, and then we have a potential packaging system for putting in either antigens directly on the surface, or perhaps for particular molecules that might infect particular bacteria and present them in the ecosystem within the gut. So and there's nothing like a few EM images just to kind of bring some kind of clarity to what's going on. So in 2A here, what we have is isolated bacteriophage and bacteria from colostrum. What we were hoping to do, we haven't got there yet, is identify bacteriophage infecting bacteria where we can co-label them with gold. But in the meantime, what we have here in 2A on the left-hand side is 2 bacteriophage, probably Podoviridae, at various stages of attachment to the probably Rheumatococcus rumens in the colostrum, and giving some evidence to the fact that even in colostrum we have this dynamic interplay between predator and prey in that environment going into the neonate. 2C, I'll deal with. First of all, this is like a positive control that we have for tailed T4 phage, where we have an anti-coat antibody, and we use an anti-murine antibody, which is 20 nanometer gold labeled, to give this kind of corona-like appearance around it. And what we have here in the middle is just a kind of first semblance of some data for phage-like particles. I'm keeping it like that in phage-like particles so that I have an out. But these are, if you like, sheep. They're 10 nanometer gold labeled sheep anti-bovine IgA antibody, so that the secondary antibody carries the gold label, binds to the bovine IgA, and then if they're co-associated with phage, then we see this on the electron microscope. And I'd like to thank McDermott for these pictures, which we're beginning to see. So this is all very good if it's one animal, but what we've done is without giving you the data, we've kind of done some crossover studies looking from, the immune profiles in one animal and seeing if they can pull out phage from another. And just to really give you the bottom line is, yes, we can. So what this seems to mean is that there appears to be some uniformity of the epitope targeting to enable inter-animal isolation of matching phage. So we're beginning to see the signs that, there's cross-individual recognition of particular phage, perhaps by recognizing the same epitopes, and then this then may give us some insights into the kind of fact that this might be a pan-species phenomenon or even pan-mammalian. I'll get to that briefly in a minute, and that we may be able to again come up with beginnings of some mechanisms as to how we get a colostrum seeded with an immune biome, and how do we get from the gut to the colostrum and figure that out. And we have some ideas on that involving maybe the lymphatic system. So it is a gut ecosystem, and you can't really look at the gut ecosystem with just looking at bacteriophage. You really need to look at bacteria. This is a smaller study on an N of 4 where we looked at the immune biome of bacteria from colostrum and compared it to the bacteria in the same animals. Again, these feces were harvested within 6 hours of delivery. So on the left-hand side here, with all the pink colors, we have the kind of classic bacteria that one would see in a fecal microbiome. And on the right-hand side, we have the 4 animals in the 4 categories: colostrum, IgA enriched and IgG enriched. And basically what you see is something similar to what we're seeing with the bacteriophage. There's not an awful lot of difference between the starting colostrum and the microbiome that you can pull out, and you can see lots of bacteria that would digest the milk proteins and that kind of thing. But what you can say is that there's a vast difference between the two, and if you look at the kind of differential abundance between feces and colostrum, there's no crossover. So from an immune seeding perspective in the colostrum, what we can say is that it's not, and we wouldn't expect it to come from the feces, but it's the most accessible of going from mouth to anus. So what we're hoping to do is get some opportunistically kind of culled animals from a herd and in the kind of post-delivery period to kind of map it out from mouth to anus, and then look at the lymphatic system as well to see what role that plays in. So the conclusions: there is an IgA biome. I can tell you there's an IgG immune biome as well. Why have it? We're trying to still sort that out, but from an evolutionary perspective, it must be important. And of course, it must be relevant to mucosal assisted immunity, the maintenance of tolerance and health. Evolution is not wasteful. We don't produce grams and grams of IgA per day, and we don't produce grams that have reactivity against a biome without having some relevance. So just to give you some sense of what we're doing in other animals, we're looking at the human. Aoife O'Sullivan is going to talk about that shortly. And we're looking at, as I said, the beginnings in non-human primates with Dublin Zoom. We have some early signals that we can isolate phage that have an IgA signal using human reagents, although that has presented difficulty for one animal. And if I could take this opportunity to just say that I have a postdoctoral fellowship position under the INSPIRE program in UCC. The closing date is the 30th of September. There's the QR code if anybody wants to kind of maybe have a look at it or talk to me later about the details of that 2-year funded postdoctoral fellowship. So with further ado, I will conclude my own talk. So any questions for the speaker?
Hi, thanks for the fascinating talk. Some studies demonstrate that in the small intestine, you'll have clearing a certain type of phages. And for instance, some cyphoviruses, you don't have clearing from Ig-like immune responses. So could you speculate why that happens to some phages that you can see them, and then some other tailed phages you don't see that clearing?
So the small intestine is a kind of fascinating environment from a kind of an ecosystem perspective. It's if you could do the comparison, we'll say, with the colon and the small intestine, the Amazon versus maybe the Atacama Desert or something, the kind of, there's not an awful lot of opportunity to meet your kind of predator prey in the Atacama. So with the small intestine. So I think lots of the studies are underpowered, my own one included. You need huge amounts to kind of really power the study, and to say that something is not present is really, it's the presence or absence really lies in kind of the robustness of the presence. Kind of saying it's not there is actually probably in some instances the lower limit of detection of what the bioinformatics can give you. So I'd say that might be an element of it. And of course, if the prey isn't there, well then of course you won't get the predator in this case being the bacteriophage. Okay, I think in the interest of time, we'll cut the questions.